Good morning, class. Today we'll be talking about DNA methylation. Now, can anyone tell me what they know about DNA methylation? Yes, you, Jessica. DNA methylation is a heritable epigenetic mark involving the covalent transfer of methyl group by DNA methyltransferase to the C5 position of the cytosine ring of DNA. Very good. Now, does anyone know why DNA methylation is important? Yes, you, Dylan. DNA methylation is important because it plays a role in a number of key processes in the body, such as genomic imprinting, X chromosome inactivation, and also, if it is dysregulated, it could lead to diseases such as cancer, right? Okay, now class, if you could look up at the board, I would explain how DNA methylation takes place and the enzymes involved in DNA methylation. These are some phrases necessary for the explanation of DNA methylation. DNA methylation is regulated by a family of DNMTs, DNMT1, DNMT2, DNMT3A, 3B, and 3L. DNMT1 methylates hemimethylated DNA in vitro and is localized to the S phase of cell division. It is the proposed enzyme responsible for copying DNA methylation patterns to the daughter strand during DNA replication. DNMT2 is a methyltransferase homolog that methylates cytosine 38 in the anticodon loop of aspartic acid transfer RNA instead of DNA. DNMT3A, 3B, and 3L have preference for unmethylated CpG dinucleotides and perform de novo methylation during development. Cooperation among different DNMTs is also required in methylating some regions of the genome, particularly repetitive elements. As previously mentioned, DNMT1 acts mainly as maintenance during DNA synthesis, and DNMT3A and 3B act as de novo enzyme in development. However, there is evidence that these work vice versa, and the functions are interchangeable between DNMT1 and DNMT3B and 3A. James, care to explain the dangers of defects in DNA methylation to the class? The dangers of defects in DNA methylation. Can I use the board? Defects in DNA methylation are closely associated with cancer, although no mutation or deficiency in any DMT has been causally linked to tumor development, most likely because of their critical role during embryogenesis. Epigenetic hallmarks of cancer include global DNA hypomethylation and locus-specific hypomethylation of CpG islands. Thus far, all examined tumor samples display global reductions of DNA methylation. Hypomethylation arises mainly from loss of methylation in normally heavily methylated repeat elements, leading to genomic instability and oncogene activation.